One of the most important insights that evolution has into the nature of patients is that patients are bundles of trade-offs. They are not machines with replacement parts that can easily be switched out. Anytime one intervenes to change one thing, there will be consequences in other things in the patient because their traits are connected through trade-offs. Trade-offs require two connections. One is a connection between the traits. The other is a connection between the traits and fitness. Now, each of these connections could be either positive or negative, and a trade-off only exists when a change in one trait that increases fitness is associated with a change in another trait that decreases fitness. One way to conceive of trade-offs is as energy allocation. So that is a physiological way of looking at trade-offs within a single individual, the idea being that there is a limited amount of energy which is coming in. That's represented here by the diameter of the pipe R, and then a fraction of it, C, is being allocated to one function, in this case fecundity, and the rest of it, 1 minus C, is being allocated to another function, in this case survival. That's called the Y-allocation model, and it was developed by Van Noordwijk and de Jong. It has some surprising outcomes. Basically, what is going on here is that if the variation is mostly in how much energy is acquired, then this would be little energy acquired here, this would be a lot of energy acquired here, lower line, little energy, the upper line, a lot of energy. Then the variation in acquisition produces a positive relationship between reproduction and survival. If, however, the variation is mostly in allocation, then there's not too much variation in acquisition, and most of the variation then is in this region here, leading to a negative association between reproduction and survival. This analysis makes clear why it is that rich people have big houses and nice cars. That's a variation in acquisition. If everybody were acquiring about the same amount of money overall, then what we would see is that people can either have big houses or big cars, but not both. This is also a reason why we think that trade-offs are expressed under food stress in organisms or under other kinds of stress, which tends to shrink the variation in acquisition, leaving the variation in allocation exposed as the source of the trade-off. Now, trade-offs can occur within individuals or among members of a family or a group. So in kestrels, clutch size trades off with age and maturity and clutch size of offspring. In Drosophila melanogaster, old females lay eggs that have higher juvenile mortality than do eggs laid by young females. So something in the mother is trading off with something in the offspring. The same thing, in some sense, happens in red deer, where the effect on the mortality of the mother is greater if she has a daughter than if she has a son, for the son moves away sooner, whereas the daughter stays closer to the mother and then competes for food. So the gender of the offspring determines the reproductive trade-off to some extent in red deer. In general, trade-offs can occur within any set of interacting individuals whose interactions affect fitness. Trade-offs can be measured by phenotypic correlation or by genetic correlations. Phenotypic correlations can exist for many reasons. Not all of them are genetic. They can be for reasons of environmental variation. Phenotypic plasticity contributes to them. Only connections among traits that have a genetic basis will produce trade-offs that have evolutionary consequences. Therefore, the most convincing way to measure a genetic trade-off is as a correlated response to artificial selection. The idea here is that one selects on one trait and then sees how the other trait responds. And if an improvement in one trait leads to a decreased performance through the other trait, then you've demonstrated a trade-off. Here is an example of measuring trade-offs as correlated responses to artificial selection. It was done in fruit flies. Now, this is a summary of a tremendous amount of information. It's 14 studies done in 5 labs involving about 100 man-years of work. In the boxes here on the left, you can see some major life history traits, such as developmental time, body size, longevity, early fecundity, and late fecundity. And in the boxes on the right, you can see some underlying physiological traits, such as desiccation resistance, ethanol resistance, yolk protein, messenger RNA, mating speed with young, and things like that. What I would like you to do is follow the arrows, and what you can see here is that if you select for increased development time, then the consequence, the trait at the other end of the arrow, is a larger fly. If you select for larger flies, then the result at the other end of the arrow is longer development time, and those are indicated by these plus signs. However, if you select for early fecundity, to increase number of eggs laid early in life, that reduces lifespan, it reduces longevity. And if you select for longer life, that reduces early fecundity. That's indicated by these negative signs here. It is a complex diagram, but the important point that it carries is that organisms are bundles of trade-offs that are connected to each other and that are not free to evolve independently of each other, and that things that we see at the level of major life history traits, like development time or fecundity, are underpinned by corresponding changes in physiological traits that make a lot of sense. Let's take a look at the phenotypic trade-off in women. Here are the number of women for whom fecundity was observed in Framingham, Massachusetts. Those are the blue bars, so you can see that ranges between about 600 and about 1,000. These are the number of children that they had per lifetime. That's CEB, children ever born, or lifetime reproductive success. This is their relative mortality risk here. Okay? So, the first 2 children, these 2, decreased the risk of death, and it then increased linearly with the 3rd, 4th, and the 5th child. And when we used the pedigrees of the Framingham population to estimate the genetic component of the variation, the genetic correlation between children ever born and lifespan was negative. It was minus 0.88, which is a very, very strong negative genetic correlation. In other words, there was a U-shaped relationship for the cost of reproduction for women in Framingham, and the estimated genetic correlation was negative and strong. And the negativity of this correlation is dominated by this portion here of the phenotypic graph. We then did a genome-wide association study looking for genes that affect the relationship between children ever born and lifespan. And we found 2 single nucleotide polymorphisms that were associated with that relationship. One is near a gene called EOMES. It's a master regulator that's also implicated in bladder cancer and in multiple sclerosis. And another is near a gene that's associated with brain development and vaccine response. In both cases, the variants that increased lifetime reproductive success decreased lifespan, so they were associated with higher risk later in life. Others have also found genes that increase fertility early in life while increasing risk of chronic disease late in life. For example, alleles in the p53 pathway. Here, mutations are probably more frequently involved in cancer than in any other gene. And here, the benefit early in life has to do with implantation success. So that's an extremely early event. And embryos that implant better into the endometrium are at increased risk of cancer later in life. Similarly, the early onset breast cancer gene, BRCA, is associated with shorter birth intervals and increased reproductive success in some samples. The ApoE4 allele is a gene that improves cholesterol metabolism, but it increases risk of Alzheimer's. So you can see that these are all places where a gene is conferring enhanced reproductive advantage or enhanced survival advantage earlier in life, but is also a gene that confers greater risk for either cancer or a neurodegenerative disease late in life. These are cases of antagonistic pleiotropy that support the evolutionary theory of aging. Now, what about hormones? Hormones are mediating lots of responses in bodies, and they are arguably a priori involved in trade-offs. Well, those trade-offs differ a bit in males and females. In males, testosterone and other androgens are regulating the allocation of energy between reproduction and immune functions. During infections, energy is switched from maintaining skeletal muscle, red blood cells, and bone density to increasing the immune response, and it does so more in males than it does in females. In uninfected males, the normal maintenance of secondary sexual characters by androgens diverts energy away from immune function and increases susceptibility to disease. For example, men who receive a flu vaccination experience a drop in testosterone levels for 2 weeks compared to a somewhat comparable control group. In brief, macho can make you sick, or it can at least leave you susceptible. On the other hand, in females, leptin is the hormone mediating trade-offs between reproduction and survival, or at least it's one of the important hormones that mediates that relationship. Leptin regulates energy intake and expenditure through effects on appetite and metabolism. It's secreted by fat cells, and it reflects overall fat storage in the body. It's a signal of body fat content. It binds to that part of the hypothalamus that's known as the satiety center, where it signals that the body has eaten enough. It inhibits neurons that stimulate eating, and it stimulates neurons that inhibit eating. When women come under nutritional stress, some reproductive traits respond sensitively and some do not, and these reactions are mediated by leptin. Ovarian function, duration of gestation, and final birth weight are sensitive to energy balance, but the rate at which energy is supplied to the embryo by the mother is much less sensitive. Pregnant and nursing mothers are strongly stressed by poor nutrition, which causes the early birth of underweight infants and increases interbirth intervals of their mothers. However, milk production is relatively insensitive to stress, and this is why even in starving populations you will see mothers whose bodies are shrinking continuing to lactate and continuing to suckle their infants. There is an interesting trade-off at the population level, and we are going to use this concept later on, which is why I'm introducing it in this introductory lecture on trade-offs. Deviations from normal, which is something you might think of as being the adaptive center of a distribution, are often unavoidable, and they have costs. Any complex process that's composed of components, each of which can vary, will be expressed as a range of phenotypes that has a central peak and it has rare extremes. So it's a distribution that has a frequent center and rare tails. If the center is selected, then this is the condition to which individuals in the population are adapted. They are not adapted to the extremes, however, the extremes are unavoidable. This is somewhat similar to the concept of recombinational load in population genetics, but in fact it's produced both by genetics and by development. It's produced by anything that causes variation in the components of complex traits. Individuals here are functioning well. They are one portion of the population. Individuals in the tails are functioning poorly. They are another rarer portion of the population. And so this is really a population level trade-off. A critical question is whether trade-offs actually evolve more slowly than do other traits. Sometimes, in fact, they evolve nearly as fast. So in bacteria, antibiotic resistance is thought to be costly. It's thought to be bound up in a trade-off. In other words, it's good when the antibiotic is there, but it slows the bacteria down when the antibiotic is not there. However, if you ask, how rapidly do the costs of antibiotic resistance disappear when bacteria are selected in the presence of antibiotics? The answer is, well, pretty fast. This is called compensatory evolution, and it can eliminate the costs of antibiotic resistance in bacteria within about 6 months of selection. Now, what's the importance of that? Well, as soon as genes for resistance no longer have a fitness cost in the absence of the antibiotic, so you pull out the antibiotic, there are the bacteria, they've evolved resistance, but it no longer costs them anything, the resistance genes have become neutral. And what that means is that they're much, much slower to disappear. They persist in the population for a long time. Now, this rapid evolution of a trade-off makes the trade-off disappear fairly quickly. In this case, it was a fairly simple trait, and it was possible for evolution to adjust it. Evolution will act to reduce costs wherever they are consistently expressed and encountered, and where reduction is possible. Now, in many cases, trade-offs persist because reduction is not possible, and in other cases, they persist because the conditions under which the costs are expressed are also infrequent. So to summarize, trade-offs are everywhere. They're the default condition. All traits are bound up usually in some kind of trade-off. For traits in a patient that are involved in trade-offs, a change that improves one condition could worsen another. For example, the negative side effects of many drugs. So you have improved symptoms for the thing the drug is targeting, but you have negative side effects. The trade-off between testosterone and disease resistance in males. And importantly, the trade-off between reproduction and survival that shapes the evolution of aging, developed by Van Noordwijk and de Jong. It has some surprising outcomes. Basically, what is going on here is that if the variation is mostly in how much energy is acquired, then this would be little energy acquired here, this would be a lot of energy acquired here, lower line, little energy in other things in the patient because their traits are connected through trade-offs. Trade-offs require two connections. One is a connection between the traits. The other is a connection between the traits and fitness. Now, each of these connections could be either positive or negative, and a trade-off only exists when a change in one trait that increases fitness is associated with a change in another trait that decreases fitness. One way to conceive of trade-offs is as energy allocations. So that is a physiological way of looking at trade-offs within a single individual, the idea being that there is a limited amount of energy which is coming in. That's represented here by the diameter of the pipe R, and then a fraction of it, C, is being allocated to one function, in this case fecundity, and the rest of it, 1 minus C, is being allocated to another function, in this case survival. That's called the Y-allocation model, and it was developed. One of the most important insights that evolution has into the nature of patients is that patients are bundles of trade-offs. They are not machines with replacement parts that can easily be switched out. Anytime one intervenes to change one thing, there will be consequences